All right. Thank you very much. I would have loved to be actually physically in place today because I've never been in any of the three Baltic states, and it would have been great to pay a visit and have a look. But that's, now it's the second best thing, at least to be virtually present. Um, Usually, many of the presentations that that I give on this topic, most of them are on publishing in general. And this is, of course, something um, about infrastructure, right? The way we interact with each other is something that should be supported by institutional infrastructure, I would argue, but that for the longest time has been served by non-institutional infrastructures that we have outsourced. And good infrastructure is a little bit like good referees. They're invisible, and people only talk about it when they're really, really bad. And this is why I, as a user, like I'm not a professional in any way, shape, or form that has to do with infrastructure um, as their day job. The only reason I'm involved in this is because it's so bad that I thought I need to do something about it. And besides just talking, some of the things that we do here locally is to try and just build the infrastructure ourselves that doesn't exist. This, to me, appears quite silly because this is sort of like um, if you move into a new lab and you have to pull all the cables for the electricity yourself because they don't have any electricians at the university. And this is a little bit how I feel. This is a little bit how it is. And I'll show you some of the things that we've done. And I hope that I can, or that you also find it quite silly that these sorts of things have to be developed by the people who produce the data rather than by the people who are employed by the institution to make sure that we can uh, create and collect data and create science and make science happen. So with this preliminary introduction, let me share the screen. So let's see, where are we? Here it is. There you go. So I hope you can now also see the screen. Yes. Okay. So it's about automating the data publication process, right? So you want to make this with as little additional burden on the researcher as possible. It's like we talked about electricity. So like go into a room, you flip the switch and the light comes on. You turn the faucet and the water comes out. This is how it should be. And or on your private phones, if you take a picture, you can easily then once it's taken, you can just go on it, click on it, and send it to whoever you want with a flick of your finger or thumb or whatever. This is how it should be, but this is not how it is. And so that's why we have started to develop um, things ourselves. On the larger scheme, the consequence of this infrastructure not existing is that data is a threatened research output. Now, this is a very old um, graph from a project from 2011, but I doubt things have changed for the better most recently. Most of the data, and maybe the percentages change, but most of the data is on some local disks and in drawers in the local institution and essentially not accessible. Then some of the data is up here with publications and data representations or in structured databases. But even those databases that are up here that seem fairly stable, that are like, or at least the stable quarter of the research data, even those have a problem. And those problems aren't really old, right? So this is a database called SwissProt. We have about 1,300 such databases in the biological sciences. And this is a report from 1996 that says that they have funding problems. And so the database would have to shut down if they don't get money. And the interesting thing is that these databases are run on project funding, not on infrastructure funding. And to this day, many, many years later, almost 30 years later, this still hasn't changed. Of those 1,300 databases, most of them run on project funding, which then, of course, means that the older the data is, the more likely it is that it is disappearing. And this is what another paper a couple years later has found out, that the availability of research data declines rapidly with article age. So you have links in articles to the data, to the raw data of the for the publication, and the older the paper is, the more likely it is that the data link goes nowhere and you can't get to the data. Still today, well, not, well, 9 years ago, there are still funding problems. And those are just slides to show you that this is something that has been a problem that has plagued at least the biological sciences for 30 years. And still today, nothing is really happening and nothing has happened. And so that's why people now are still, even though the problem had persisted for 30 years, um, are like wondering, oh, okay, maybe we should have done something now that we have actual enemies of science being in control of the funds, of those project funds that run the databases, right? So, oh, maybe now we have to do something to safeguard our data. When Trump takes a giant wrecking ball to US research, how can we safeguard, again, the word safeguarding, against a Trump data dump? The desperate rush even, right? So 30 years, who would have known that our databases are in jeopardy, right? So 30 years of knowledge, and then all of a sudden, oh, we have to rush to save data because all of a sudden, those 30 years are gone in an eye blink, preserving the federal data. And for me, as a person who works on Drosophila fruit flies, the most threatening development is to a database of fly genes, namely called Flybase. And without that, I can actually stay home. If that really shuts down that database, I can't work. So for me, you can, and people like us who work on fruit flies, essentially, you can shut down all publishers, all subscriptions, all anything, the literature. We can somehow continue to do our work. But without this Flybase, without this database, we can't work. This just means the end of it. And so this was 30 years in the making, and people have been warning for this for a very long time. And so here I would have to say the scientific community only has itself to blame for its inaction and its vulnerability to such a situation. So that's why we have started early on in our lab to try and think of how we can do this in a way that is decentralized, that is automated, such that there's no individual that can take down a large portion of the scholarly research data. And so we're looking at this base of the pyramid from the previous presentation, or previous slide, from one of the first slides, that the data do not remain longer on disks and in drawers. And for all of you who work with digital data, probably for a lot of you, the workflow looks a little bit similar to this. So you have an experimental design that produces metadata and data. Sometimes these are in the same file, sometimes these are in different files. This is really a little bit different in research culture in how this is handled. But then from the data that you produce, you have a quality control process, and then you use the data and the metadata together to generate, graphs and stats about whatever it is that you're interested in. And then once you have sufficient of those results, you write a publication, and then someone asks, well, can I have a look at your data? And so this is the usual process of why this is working. And so one of the first projects that we did now, quite a lot of years ago, don't even remember when that was. It was probably 15 years ago or something like this, 10, 15 years ago. We worked together with our Figshare, and that was before Figshare was bought by Digital Science, and Digital Science is a sister company of Springer Nature and the daughter company of the publishing conglomerate Holtzbrinck, to deposit raw data and statistical evaluations automatically without any additional work. And so what we did was we created scripts that used R and R Figshare to take the metadata, the quality control data, and the stats that are being generated by the scripts that we also use R, to take all of those and put them in a database right away. So whenever the researcher generates one of those graphs, which the researcher has to do anyway. They run the R script, for instance, and use the evaluate the raw data into these graphs. Whenever that's being done, the data is being updated. And then from those data, you can generate your own research report for your lab meeting, or you can make a paper out of it. And the way this then looks essentially is that you use our Figshare, you have Figshare tokens, you just get an account on Figshare that you use, and then essentially that's in terms of the general idea of how this works is you just enter this into an R script, and whenever you call the R script to do what you actually want to do for yourself, namely generate those graphs and evaluations, in the background without you seeing it, it uses Figshare to update the data set that you're currently working on. So that was the first thing that we did, and we do that. We could, well, we're not using this anymore, but at the time we used this for a while for a subset of the experiments that we're doing. Another thing that we tried to initiate together with F1000 Research, that was before Taylor and Francis bought F1000 Research. You see all the things that really work in science then get bought up by the big corporations. Was something that F1000 Research called living figures. And the idea here is that why should we give the readers a static version of our data? Why shouldn't the users also be allowed, the readers be allowed to slice and dice and look at the data in all kinds of different ways? And so we just supplied with one of our publications, and that's the one if you want to have a look at it. We just supplied the publisher with the raw data and some of our evaluation scripts and said, yeah, there you go. Just use that and let the reader decide what they want to look at. Clearly, this was just a proof of concept. So for instance, the way we did this was, let's see here for figure 3, for example, if you click on figure 3, it says loading, please wait, because it loads the data and then uses scripts to display them, for instance, with a Tukey plot. So you want the whiskers to be displayed like this, and the user can also go up here and from the pull-down menu pick another depiction. So here it says that the whiskers intent to value is 1.5 times interquartile range from first and third quartile. You can apply or reset. And then if you use a different one, 10 to 19th percentile, the whiskers now is sent from the 10 to the 19th percentile. And dots are the value above or below the whiskers. You see, it gets plotted a little bit different. And this is just, as I said, just a proof of concept. You can imagine all kinds of things in which you can display data differently for people who are interested in this. And why should the authors have full control over that sort of thing? A final aspect in this paper is that for all those that use the same experiment, they can actually add their data to our data. So we compared certain fly strains. So those are the ones here shown in green. It doesn't really matter what kind of a plot this is. It's just simply that we have used these 5 strains and characterized them in a particular way that they show up in these places in these 3 plots. And then other users, in this case, 3 other users, can after publication of the paper can say, oh, this is interesting. I wonder how my fly strains look like in this evaluation. And what you can see is that their strains are actually quite different from ours as well. Not in this plot, not from this perspective, but in these 2 perspectives, one can actually see that these come to lie outside of the area that our fly strains provide. And this was a quite interesting insight that this was indeed possible, that our fly strains don't cover the entire range of possibilities. And so if this is, you notice something in your data, you wonder how general is this? Do other people find this too? Then you can do this in this kind of way. And this, of course, has direct effects on reproducibility, right? Because it shows, well, can other people reproduce the same data or the same results that you have gotten, or does something else come out? Or is it something more nuanced in that, they're somehow similar, but they're also different, and we can actually quantify the difference and can find out where the difference is and maybe find out what the reasons for those differences are. So that was the second aspect. And now the one I want to talk most about 15 minutes in, is the current project that we have just essentially finished. There's a few things left to go. This is where we automatically link the raw data that we produce, the data models that describe the structure of the raw data files, the lab journals, and the publications, and to automate as much as possible of this. These right now we use YAML markup languages. Essentially, we use YAML, XML, and HTML. But you can use JSON, you can use whatever you want. The HTML we mainly use for output, so this is useful because it's browser readable. People can just browse and click through the results. But all these things are not necessarily tied to these kind of file formats. Essentially, well, let me look at or emphasize first that this is a collaboration with two colleagues in the library, Dr. Gernot Deitzer and Konstantin Lehnmeier. They're helping us design the project, and what they do is they implement the automated synchronization with the publication server of the library. So the general concept from our end, so before we go to the library, the general concept is that we have a lab journal that's both human and machine readable. That's why we use YAML, and you will see an example of such a YAML file. Because I'm currently doing experiments, I'm just taking a short break right now to give this presentation. I'm doing experiments right here. And so I can show you the kind of data set I'm working on right now. And I keep notes in a machine readable and human readable lab journal. In that lab journal, I note down which of those raw data XML files that each experiment is generating belongs to which experimental group. Let's say I have 3 experimental groups, and file number 1 goes into group 1, file number 2 goes into group 2, file number 3 goes into group 3, and then file number 4 goes again into group 1, and so on. So I have 3 groups, and I have to let the evaluation scripts know which file goes to which experimental group. So those are the raw data. Then our R scripts that we use for this project or for this data set, these R scripts then we can read the YAML file, that then they know which of these XML files goes into which group, and they do 2 things. These R scripts do 2 things. At first, they generate quality control output such that humans can click through the results and see, okay, this file needs to go, this file needs to go, and this file needs to go. And then what happens is you just comment out those files in the YAML lab journal, but you don't delete the files, which means that if you change your quality control criteria, you can also change the output that you get essentially in this quality control step, right? So if you say, well, the fly must behave in a certain way in order to be included in the evaluation, and then you find out that, oh no, this is actually was a wrong idea. We can include those files, but other flies need to be excluded. You can still do that at any point in time as you're learning more about what it is that your experiments are actually, what they actually mean. And then the second thing that the R scripts are doing with the YAML and the XML files is they create a single evaluation sheet where, for instance, the statistics are being done between those 3 groups, for instance, these sorts of things, right? So where you ask the scientific questions and then give results to those scientific questions. The raw data themselves, they contain DOIs to the software that is there for the data collection, the software that we use for data analysis. So these R scripts are also linked here where they are. And there's a link, a DOI, to the data model that describes how to read these XML files. So both the description of the XML files is linked and the software, which means that anybody who wants to see how we created these, how these results happened, can download these different sets of software, learn about how we do this, and then recreate the exact same results that we have recreated, or change the quality control criteria and see if that changes the results, or if the results are stable against these quality control criteria. And then from this HTML evaluation sheet, in the ideal case, if we would have a decent scholarly infrastructure, which we don't, that's why we are all in this workshop. What you could be doing is you can just drag and drop, let's say, such a figure that's being generated in this from this HTML document. You can just drag and drop this into your authoring environment, and via this drag and drop, ideally, this does not happen yet, but this could happen. You would automatically link back to this entire project. So a single drag and drop operation would allow every reader to access all of this, your lab journal, your raw data, your quality control results, and your evaluation sheet that contains many, many more evaluations and analyses than just the one figure that you put into your documentation or into your publication. Just a single drag and drop movement would allow you to do all this. And because it is automated, you cannot make any copy and paste mistakes or any other things such that the link would eventually be broken. So if you do this, if we were all to do this in that way, then we would never have any of those data problems anymore. So how then does this get published? On my local computer right here that I use when I'm talking to you, we have the YAML file, the raw data, and all the other things. And whenever I log in, this is one of the events, I can define on which event this would happen. We run a Python script that then checks and compares the corresponding folder on the publication server of the library with my folder here locally. If there is a difference, they get copied such that the publication server always mirrors the raw data folder. And that then means we have a linked situation or a linked document that links to all the other files and documents that are necessary for later reuse for someone else, to use in a publication, to use for whatever. For me, as a PI, it's very useful because if everybody then in our lab is going to use this now, from now on, actually, because it's fairly recent that we finished this into production stage. So from now on, everyone in our lab will be using this. And so if a bachelor student comes, does a project and leaves again, I know precisely where the data are because I don't, because I sort of say we wrote the script such that I can always find it. And I don't have to tell the students that are only here for a few weeks or the graduate students that are here for 3 years, oh, please make sure they can access your data and find your data. No, it's all automated. And the data are all in an easily findable place and very structured. So with this, I would like to show you an example. There are going to be 2 examples. The other one I will be pasting into the chat box momentarily. For those of you who want to have a look an early look, this is a completed data set that I collected. What does it say here? In March. And you can access it here under this URL. And then if you click on this HTML file, and we will go through an example of those, then you can click through all the other files that I linked to already. All right, so this is just very briefly. Now for the last 20 minutes, I'll just show you the files that we use. This is just very briefly an introduction into the kind of efforts that we have made over the last 10, 15 years and how we're currently thinking about this. And for you, an example. I would like to now give you the opportunity to ask a short questions. If there was anything in here that wasn't perfectly clear, before I would then go on, so maybe for 2 or 3 minutes, before I then would go on and show you the examples of the files that we use in this project. And of course, if everything was perfectly clear and nobody has any questions, then we'll just move on to the files.
I just had one question. If nobody else has a question, do you maintain, so with each of these updates, do you maintain a version control, and where would that one live? Does it live on your lab computer, on the publication server, or on the both?
All right, so right now we do not have version control because the way these experiments sort of work is that you need to reach a certain sample size.
Okay.
Right. And so for these experiments, for most of these experiments, we have a fixed sample size, which means that we collect data every day. And unfortunately, fly work is fast, but not that fast. We still need several weeks to collect data to reach that criterion for sample size. And And then the data set is usually finished. And the only thing that then can happen is that reviewers, upon publication, they say, oh, but you need to add a control group, another control group, or you need to have a higher sample size. Or they say, oh, your quality control criteria were insufficient. You need to throw these and these flies. And then we drop below our sample size criterion when we throw out those flies. So we have to measure new flies. So that sort of thing can happen. So that's why version control of the DOI, for instance, that each data set then gets, is not high on our priority list, but in fact, it's an issue that we've now added to our GitHub repository for this project, that we will add versioned DOIs as well, such that users and readers can go back and look at, okay, what was the version when we submitted it? For instance, when we submitted it, what was the version after peer review? And these sorts of things. This will then be possible via the DOI, the versioned DOI feature.
I just, because technically I'm a little bit, weak. So I just have a question. Is it within the lab? Is it within the institution? How do you manage the size of the amounts of the data over the years? Because you're going to do it for many years. And who takes care about it? Is it the library? Who's doing it?
Yes. So this is just for now, this is just my laboratory. And the data we produce are really tiny data. So I'm doing this right now. I could go to my data archive. This is over the last 30 years. This is all the data that I personally generated and some of the data that other people have generated. And I look at all of the size of the data archive over decades. And it's only 130 gigabytes for now, right? So you see, these things are tiny data. So the amount of data in this case for this kind of project in our lab is tiny. So this is not an issue. But of course, it would be if other people use it. I will show you what the criterion is. So you can use that sort of thing. The concept can be used on any data. And now if I show the data files, I will explain why this can be used on any data, not just our kind of data.
So this is not done for recordings, because we record behavior, and that's like half an hour video, that's 1 gigabyte. And we do lots of them. So how would you manage that?
Oh, so what we do, right. So what we do, I mean, otherwise the size of the data, of course, has to be commensurate with the possibilities that your institution has, right? So if this is an important project and the data needs to be stored for a certain amount of time, well, then the kind of resources have to be made available. Just like if I need a certain amount of electricity, I need a certain amount of water, I need a certain amount of gas, that needs to happen, right? This is part of the infrastructure component, right? I mean, the people here next door, they have 2 nuclear magnetic resonance imaging machines there. That's what they need. That's the infrastructure they need. And so that's what needs to happen. The way we record our behavior here is that we have a recording device that records in 12-bit at this point at 60 hertz. So that's why you only get, for each 20-minute experiment for a fly, you only get like 1.5 megabytes of data, because we don't store the videos. Even if we have some video-based experiments, and usually what we do there we extract the relevant parameters online during the experiment and never save the video, just the extracted data.
Okay, I let you move on. I let you move on. Please show us.
All right. Okay. So the first thing I'd like you to see is the YAML file, like our lab journal. And now I need to see, check where it is. No, that's not the right one. I think here, this is what it is. Oh, no, before that, let me go to I have a browser window. I think that's what it is. Yes, I'll share the browser window and I paste. You see the browser window here. This is a project. This is a different project. It looks the same, but you see it's a different project. This is the one I'm currently working on. And I will, let me see here. Where's the chat box? Here's the chat box. I will paste that in the chat box. It makes it easy for everyone to just click on. Then everyone can look at this. And so what you see here, then if you click on this HTML, you see a lot of data already. You see, right? You see graphs. Doesn't matter what it is, but you see lots of things going on here. And we'll get back to this browser window in a second. Let's first look at the YAML file that the Python script is reading to synchronize the data. And I think that was here. Yeah, so now you can see this. I hope this is big enough for everyone. Let me just, I don't know what your screens look like, so I'll just make this now really, really big. At least on my screen, this is very big text now. I hope this works for you as well. And the way this is set up is a typical YAML file, which means that you have these tags that belong into a different to one block, right? So you can see you can open and close these blocks, and the file starts with metadata information and then resources. Those are the two. There's some other groups further down, but those are the two main ones. The metadata contains information such as institution, department, subject, data type, open access type, funding, licenses, author, and experiment. And usually this consists, it's a very stereotyped structure. It's loosely took inspiration from the frictionless data format, which is usually JSON. But otherwise, I mean, it doesn't really matter what markup language you use as far as I'm concerned. And so what you see is that each block has a name, a title, a description. The name is one that you can use, you could in principle use to generate file names from. The title is as it says, the title, and the description is just a longer description of what this block entails. And then usually an ID, in this case, a ROR ID for the University of Regensburg, or other kinds of IDs to disambiguate anything that's in name, title, and description. And so you see this is very similar. The structure is very, very similar throughout those different blocks. And so the author, you have the name, then the description, the first name, the last name, and an ID, in this case, an ORCID. And then the machine that you saw here next to me is a torque meter. For this particular torque meter, we have just also recently developed this one. I haven't generated an RRID yet, but here you would put an RRID for this measurement device in there, for instance, or for the experiment that you're currently using. A name, a title of the experiment. So here we manipulate neurons in a structure of the fly brain called the ellipsoid bodies. It doesn't really matter. And then, yeah, we have these kinds of changes in these neurons in this experiment. And here, again, we have a description of the experiment that's more verbose. And for instance, where you put databases, I talked about the fly-based database that's so important for us. Here you put the IDs in there of the fly strains that are listed in these fly-based database. And so the only thing that the script that synchronizes our project folder with the publication server does is that it reads some of this information, usually, actually, now that I think of it, probably most of this information, right? It just reads the metadata and then creates the page that I just linked to, where you then can click on the HTML document, right? So it reads this information and this kind of metadata, and that's sufficient, which means that anybody who has a structured metadata file and then some other data can use that sort of process, because it doesn't really matter what comes afterwards. This is what so for the automated data publication, it doesn't matter what comes afterwards, right? The script reads this, uses this information to publish the whole data set, excuse me, publish the whole data set on the publication server. Right, so that's the only thing that's really required generally. It's generic, and you can use it for any experiment. This is in no way, shape, or form specific to the kinds of experiments that we do. And then what we use here, this is the resources, is when we have the three groups, for instance, in this case, 3 experimental groups. I do not know which fly strains I'm testing. I just get colors. So this is the red fly group, this is a green fly group, and the purple fly group that just have purple stickers on their containers. That's all it is. That's why they have colors. It's not that the flies have colors, it's just the containers of the flies have my technician put colored labels on them. And you see, those are the different XML files. Each file represents one 20, 30-minute experiment, roughly, with a fly. And then there are notes to each fly that help me when I do then later, I haven't done that yet for this data set. When I do quality control, it helps me look, okay, at these flies, I need to look in particular. Here, when I look at these data, what these remarks tell me that this experimental group is useless for a particular kind of evaluation, which means that probably the whole data set for this set of evaluations is probably going to be useless. But I can use other evaluations for other evaluations. This is still work. So this is, so to say, the lab journal part where you can note down everything that had anything to do and that happened during the experiment. And then finally, at the bottom, we tell the scripts certain things that are of relevance to us, but that each user can set. So we set 3 significant levels from 0.5% to 0.1%, to 0.01%. We set certain Bayesian priors. Users can pick the colors for the graphs that they generate. And then we pick certain components of the experiment. And those are the ones that with which we want to do experiments and statistics. And we have 3 at the moment. Those are the most common ones. We have 3 different types of statistics where we test a single group against 0, where we test 2 groups against each other, or if we have 3 groups, as in this experiment, we test, or in this data set, where we test all 3 groups always in dyadic pairs of tests. And then in the end, again, here we have, title, description, data. Data just says, do we want to do these statistics or not? So yes, all 3 single groups should be tested against 0. No, we have more than 2 groups. There's no sense in testing just 2 groups. So this is 0. But yes, we have 3 groups. All of them should test it against each other in a 2-sided test. So we say here, yes. And the only thing that the script does, the Python script that synchronizes the data set, the local data set with the publication service, that adds this electronic publishing ID, the 77639, which if you look into the chat box is exactly part of the URL and generates the unique identifier for each data set. So this is what the YAML file looks like. And this is what I call, this is a lab journal that has 2 roles, 2 simultaneous roles. We use it as a lab journal, so it's human readable and human writable. And because it's a markup language, it's also machine readable. So we can use it for various purposes, such as guiding our R scripts to which data file has to be evaluated in which experimental group. And it can be used for, again, machine readable purpose of synchronizing the data with the publication server. Now, finally, in terms of looking at the content of the files, here you see an XML example file of one of the last experiments I did yesterday, where you see that again, you have metadata, a metadata field, then you have an experimental sequence field, and then the time series where all the raw data actually sits. And you see that in the metadata, we link to a license. Then here are the 3 DOI for the recording software, for the analysis software, and for the data model. So if you would take these DOIs and type them in, you would get to these software packages of the data model. Again, this is the person who did the experiment with their orchid. Then the name of this particular fly that we used. Again, experiment type, date and time of it, the duration of it, a description, the sampling rate, and then the kind of experiments that we used. These are just generic names. The sequence here means that because this is a time series experiment, which means that it lasts for 20 to 30 minutes. And there are different periods in this time where the experiment changes in different phases of the experiment. And this is defined here. We'll go through this very briefly then when we look at the HTML output as well. And then it just says here what kind of comma-separated variables do we use and which are the variables. You can see those here. Variable number 1 is time. Variable number 2 is so-called angular arena position. Variable number 3 is torque. And variable number 4 is the period number of that experiment. And then you just have these 4 columns here of these 4 variables. and that goes all the way down. And this goes all the way down to, in this case, 16 in total. If we start with 0, 17 different periods, where then everything else ends. And so from all of these data, so we stop this share and open the browser again and share the browser again. So from this data, our R scripts generate, so where is it? There we go. Right, so we're back to the browser from this data set, from this YAML file, right? So now if you click here, I right click on it in a new tab, I open this, you see the YAML file. So if I go, how is it? View page source, you see that's the YAML file I just showed to you, right? But now this is directly from the publication server of the university, but it's the same YAML file, you can see it. It generates one big experimental evaluation sheet, where then the three groups, red, green, and purple, with all the different flies in their fly experiments are listed. You also see this experimental sequence. You can find this here such that each individual period is shown as what we have done with the flies in this period. You can go to via my org ID, you can go and get to where I am, right? So you can find this here. You can also see, for now they're not linked yet, which is also something we want to do. You see those fly base ID right here prominently in the description. So you can go and check which flies were used here and which fly types were used here in this experiment. And so here you can get to each individual fly. Let's just pick the first one, red 00. You open this and you get to the quality control sheet, right? This is what you see here, single fly quality control report sheet for each individual fly. And so here you get into this XML file. Again, if you click on view page source, you see that this is this kind of XML that I just showed you, only that this time it's on the version on the publication server. Right? And so this is all linked here. Again, the ORCID is linked, and the other data, the other metadata that we have here is all shown. And the nice thing with these HTML documents is that you can look at the raw data. So in this case, the blue trace is the one that's really important. The red trace for these experiments is irrelevant. And you can zoom into the data and you can go and look at what the fly was doing, which is what it has here in blue, right? And you can look at this in various ways. This is not really important. This is just for us. This is important. And this helps us decide, is this an experiment that passes quality control or is it not? And so you see different kinds of evaluations of different traces and different parts. You see here fitting of parameters and extraction of parameters. These all tell us, help us in deciding whether this is an experiment that ought to be included in the evaluation or not. You see a power spectrum, a spectrogram, whatever these things means. They don't mean anything to you, but they to me tell me something about how the fly behaved during the experiment. And the same is true with here. So this is what we use to kick out flies or to keep them in. And when the only thing that we do when we kick flies out of the evaluation is we put a comment mark in front of that fly's name. And now finally then, if you go through these three groups, you get evaluations for the group red, you get evaluations for the group green, in this case histograms, evaluations for the group purple. These are different parts of the experiment that we're evaluating here to see are green, purple, and red somehow really different in some way. Over here, what you can see is that the red ones, this is a force measure actually. The blue ones seem to be stronger than the other ones, whatever that means. Since I'm blind, I don't know. In my mind, I would say that these flies, since they're stronger, should be my control flies, but we will find out. And we have the parameters that we fitted that I showed. We show them here so one can go through this. And then we have performance plots in different ways. So we have means and standard errors. We have box and dot plots with or without notches, violin plots, And then in the end, as I showed, those are the things that you specify in the YAML file at the end. We do statistics, single group statistics against 0 of those 3 groups. None of this is really, well, these seem, well, none of this particular at our level at 0.5%. With these low sample sizes, we usually shoot for 20, 30 sample size for between 20 and 30 in different ways. So here we have a box plot, here we have rainbow plots, and then tests between the groups, which are all non-significant at the moment, and then tables with further statistics on our evaluations. So you see, all of this is linked together. And ideally, if now, let's say we would put, we could drag and drop this figure into a publication. Ideally, the publication would have an automated link to this whole data set, and via this particular HTML file, you would get access to all the other files in the data set. And all of this would be totally automated. Now, with this tour de force through the different files, I'd like to thank you for your attention. And I already see there's one question in the chat box. Have you considered PID, NST for identifiers, or are RIDs easier, more fitting for your experiments? I'm not aware for PIDINST, for PIDINST. I thought, PIDs for instruments. No, I was not aware of them. I will look into this. If you have a URL, I can easily find this. Our RIDs is what I've come to know, what we find in the publications that I read. And I have not seen such IDs yet. And absolutely, if this is something that we can use and is something that's that a lot of people use, that's going to be around for a while, I will absolutely use that sort of thing if this is better or the same, at least as those RRIDs. Absolutely. Thank you. Very cool.